after this nice introduction of Henry into the fantastic world of opossum, I would now like to come up with an application referring to the transcriptome and the epigenome of the aging and the diseased brain. So concerning the healthy brain, I will focus on the prefrontal cortex. And here in this nice animation, you can see the location of the prefrontal cortex of the left hemisphere of the brain. So the prefrontal cortex is one of the last regions of the brain to mature, and it is responsible for complex cognitive behaviors, personality, decision making, and also weights the outcomes of all our actions. So for transcriptomics, we analyzed 44 prefrontal cortex samples of different age. So we divided our samples into different age groups, namely newborn, baby, infant, teenager, adult, and last but not least, also late adults. So first of all, we took a look at our transcriptomic characteristics, such as the variance of the data and also the absolute expression. So here in those box plots for each box represents, for example, the variance of each age group. So red the newborn and blue late adult, for example. And you can see that the variance of the expression data is highest for newborn and for late adults. And for intermediate age group, the variance decreases. And for absolute expression, we observe a decrease of expressions during age. So Henry already told you how we obtained those beautiful, colorful images for each individual. And here you can see the summary of our transcriptomic data. And we also calculated for each age group one mean portrait here depicted. And next we were interested in the similarity of the samples. So here you can see two representations of similarity analysis based on correlation. So for example here you can see the correlation net, where each dot represents one sample of this transcriptomics cohort, color-coded according to the age group. And if two samples are highly correlated, meaning higher than 0.5 correlation value, then there's a gray line be drawn. And here on the left-hand side, you can see the pairwise correlation map of all samples, ordered according to increasing age. And red means high correlation, while blue means anti-correlation. And you can see there that the red clusters form along this diagonal, meaning that neighbored age groups are highly correlated. And in both of those similarity analysis representations, you can see the trajectory of aging pointing from newborn to late adults. So next we were interested in the functional context of our age groups, and therefore we collected all those interesting red overexpression spots, and we already talked about this, and we collect all those red spots into one overexpression spot map, and then we cut out those red spot modules again and label them with capital letters ranging from A to G. And if we now recall those mean sum portraits of the age groups, you can see that those mean portraits are quite age specific. So if you take a look at those red spots, you can see that this red spot rotates in clockwise direction. So for example, for newborn, we detect one overexpression spot in the top right corner of the map, while for teenagers we detect two overexpression spots in the bottom edge of the star map, and for late adults we detect two overexpression spots on the left corner or left edge of the map. So in the middle row, middle column, You can now see the expression profiles of those spot modules detected. And this means we collect, for example, for spot A, we collect all genes belonging to spot A, and for each sample we calculate a mean expression value, and each bar here represents one sample. And for example, for spot A, you can see a decrease of mean expression value. So then, using gene set enrichment analysis, we were able to put those spots into biological context. So we found, for example, that all gene sets related to neuron production are overexpressed in newborn and in baby. And this finding is in line with the fact that in baby brain, the brain cells, namely the neurons, are overproduced, and also the connection between those neurons, namely the synapses, are also overproduced. And then, for example, for spot F, we see that this spot is overexpressed in adult or late adult samples, and we find that a gene set related to brain disease, namely Alzheimer's disease, pops up. So now, out of our 10,000 gene sets that I picked out some gene sets that are obviously related with brain development. So, for instance, the gene set called learning. So this gene set contains genes that are all related to learning, and we can see that those genes are overexpressed in newborn, baby, and infant, so they learn a lot. And then the expression dramatically decreases with increasing age. This is also found for cognition and also for memory. So we found that aging brain doesn't only change the structure of the brain, but aging of the human brain is also notably accompanied by cognitive decline, as seen in our transcriptomic data. So now let's switch to DNA methylation. So for DNA methylation, we analyzed 65 prefrontal cortex samples that were obtained from post-mortem human brains from non-psychiatric controls. So unfortunately, we didn't have matched samples, meaning that we didn't have both one transcriptomic and one DNA methylation sample for each patient. But we wanted to compare this DNA methylation data with the transcriptomic data shown before. So therefore we only considered Caucasian samples, and again we divided the DNA methylation samples into the already shown age groups. But as it is known that regarding DNA methylation, the fastest changes occur during the prenatal period and then slow down markedly after birth and continue to slow further with aging. We also considered an age group prenatal in addition to our already considered age groups. So here you can see the figures that I already showed you for transcriptomic data, but this time produced with DNA methylation data. So here you can see the mean age group portraits of DNA methylation data. And again, they are characteristic for each age group. And you can see again that this red spot rotates with increasing age. And here in this pairwise correlation map, you can basically detect two big red clusters. So one beginning with prenatal samples till the age of 10, and then there's a switch, and the other samples older than 10 are also clustered. So it is also known that prenatal tissue is dominated by demethylation, and then this leads to an increased methylation state with increasing age. And this finding is also depicted here in this box plot. So you can see prenatal samples are demethylated, and then methylation increases. Now I wanted to combine both the DNA methylation and the transcriptomic data. So one way I did this is with the help of those two heat maps concerning biological process-related gene sets. So on the left-hand side you can see a heat map for gene expression, and on the right-hand side the same heat map regarding DNA methylation. Again, the samples are sorted with increasing age. And if you take a look at those heat maps, you might see clusters. So I divided those biological processes mainly in three clusters or types. So the first concerning learning, then another cluster related with metabolic processes, and the last one negative regulation with growth or neuron differentiation. So you can see that there's, for both DNA methylation and gene expression, a switch at the age of 10. But now, as it is hard to interpret those two heat maps together, I transformed those heat maps into correlation plots. So again, for each biological process cluster, you get one correlation plot. So on x-axis, you can see the mean methylation rate of all gene sets regarding learning, and on y-axis all the respective mean expression value. And for learning and metabolic related processes, you can see an anti-correlation of methylation and expression in healthy brain, meaning that, for example, all gene sets related with learning are hypomethylated in young brain and overexpressed, and they then get, with increasing age, they get hypermethylated and underexpressed. And for this negative regulation of growth, we observe a correlation between methylation and expression. So now, last but not least, for our healthy brain, we were also interested on gene sets related to new degenerative diseases, such as Alzheimer, Huntington, and Parkinson's disease. So here in the scatter plot, you can see the position or location of those genes within the gene expression zone map and also DNA methylation zone map, followed by the gene expression profile and the respective DNA methylation profile. And in the last column, again, this correlation plot. So if you now take a closer look at the DNA methylation profiles, you can see that that those gene sets related with brain disease are hypermethylated in young brain and get hypomethylated with increasing age, resulting in an overexpression. And that leads us to the hypothesis that this protective and repressive DNA methylation mark gets lost with increasing age, and hence the brain gets vulnerable for brain disease. Now let's switch to the disease brain, namely the low-grade glioma. So I think also Hans already showed this slide. So in our disease cohort, we had 122 patients. So of all patients, we got one transcriptomic and one DNA methylation sample. And the results, or the cluster of gene expression data, is shown here. And for DNA methylation, you get the results here. So the gene expression data was stratified into 8 expression groups, E1 to E8. And for DNA methylation, we got six subtypes, M1 and 2 M6. And I don't want to go into detail with the subtypes, but those subtypes are also driven genetically. So for instance, if you take a look at this bar here, IDH1 mutation, you can see that E1 and M1 only contain IDH wild-type samples, while the rest of the subtypes are IDH1 mutated. So now I want to get a connection between the healthy brain and the diseased brain, and this is one way we could do this. So we got all those modules of the low-grade glioma methylation data, so those spots, and used them as new gene sets. And then we mutually mapped them into the healthy brain data, so gene expression and DNA methylation of healthy brain. And for example, for the spot module D, we found that in newborn or baby or prenatal samples, this spot is dominated by hypomethylation and then gets hypermethylated with increasing age. So for this gene set, we observed a correlation between methylation and expression. But this way is quite tricky to interpret the results. So another or better alternative to combine two independent data sets was already introduced by Henry before, using the extensiosome. So here again you can see the mean portraits obtained by single-zone training of the DNA methylation data of healthy brain. And now we were interested also in low-grade glioma, so therefore we used this extensiosome, and we used both data sets to train the some. So we took the healthy brain methylation data as it is, and we then downweighted or passively trained the low-grade glioma data. And this means that we now can directly compare the healthy brain and the glioma brain samples or the portraits obtained. So if you now compare the first row and the middle row, meaning the mean methylation portraits of single-zome training with extension-zome training of healthy brain, you can see those are identical. So this extension-zome training didn't affect the sum structure. So now we can directly compare those glioma samples with the healthy brain, as I already said. So for example, for M1 and M6, we detect an underexpression, namely a blue spot in this bottom right corner of the map, and meaning those two subtypes resemble the young or prenatal and newborn brain, while the rest of the subtypes, namely M2 to M5, has an overexpression or red spot in the bottom right corner of the map, which means it resembles the adult brain. So we can also stratify this low-grade glioma according to age. And this finding is also supported by this pairwise correlation map. So in this pairwise correlation map, you can see the pairwise correlation of healthy brain samples here and of low-grade glioma samples there. And here, in this excerpt, you can see the pairwise correlation between healthy brain and low-grade glioma. And you can see that there are two red clusters, meaning that M1 is highly correlated with young brain, and the same for M6. And for M5, for example, we detect a high correlation cluster with adult brain. This correlation map can now also be transferred to this river plot. Again, you can see that M1 and M6 most closely resembles the prenatal and newborn samples, while M5 resembles adult and late adult samples. And this means we conclude that M1 and M6 back transfers the methylation state to a healthy young brain. But last but not least, here are the survival curves of those low-grade subtypes. So here you can see the probability of the overall survival in terms of years. Here is the survival curve of M1 and of M6. So you can see those two subtypes that most closely resemble now the healthy young brain has the best, the worst prognosis. So and with this slide, I would like to summarize our two main findings. So the first one was, despite there's an overall increasing methylation state with age, so shown here in this box plot, brain disease gene sets lose the protective methylation mark with age and get vulnerable for brain disease. And also we found for this low-grade glioma data two subtypes which are being back-transferred to infantile methylation signature, and it has worst prognosis. So I would like to thank you for your attention.